Hi, this is Miss Litton, and these are my wonderful 7th period AP biologists. Say hi. Hi. We are very excited because today is Friday. And it is That's right. Okay, and we want to make sure, because I didn't do that willfully, obviously, by the 7th period sport calendar, because I was coming in on Monday. But your quiz is when you come back, right, on Monday. So we want to make sure we have... on Tuesday when you... See, I still haven't learned. Tuesday when we come back, so you want to make sure you got this down, right? And I'm also going to recommend, are there any essays related to this? Yes. And did we have a specific debrief related to it? Yes. That debrief sounds like a question I would ask on a test to me. I don't know. Sounds like a good question. I'm just saying. Okay, so make sure you do your debrief. So the very first thing we talked about was the cell theory. And that first part of cell theory, see if you can guess it, all what? All living things are composed of cells, right? If it's alive, it comes from a cell. The second part, do you remember?
All cells come from
we said, we related them, structure.
Oh.
Yeah, so cells are the structural and functional unit of, there it is, of organisms. Okay, and number 3 is that cells only come from pre-existing cells. Perfect. Then the next big thing that we spent a lot of time talking about was that cells are small. And we wanted to understand, and again, I apologize for the font. You understand, they updated my computer. I have to keep saying that. So I really apologize that it's messed up. Okay, but we want to understand why cells are so small, and we know cells are so small because they need a very large surface area per volume inside in order for them to function. Because all what?
Transfer.
Give me an E word. Exchange takes place across the cell membrane, right? And so if all the interactions take place across the cell membrane, and we talked about the Axe example, right? If we sprayed Axe in this room versus the gym, where would Axe have the greatest ability to make us run away from it? In this room, right? As opposed to the gym, yes? Because it's so much bigger, and we couldn't, if that was some sort of chemical communication, it would be hard to communicate with a cell that was that big. And we talked about the issues with eukaryotic cells, because eukaryotic cells are larger, so they have less term, so they need to have adaptations to overcome that in their largeness. And we looked at things like they have, what do eukaryotic cells have that prokaryotic cells don't? Organelles, right? And so those membrane, like the nucleus, the membrane, by definition, the membrane-enclosed organelles will help them to compartmentalize and do a job like within the mitochondria to handle, so those chemicals for that job are just in that one location and they can still function. Because if it was spread out through the whole cell, it probably wouldn't be as effective. Okay, and we'll talk about some of the membrane potentials that have to take place and why that is so critical. So we need a large surface area per volume. Are you going to need to know how to calculate that on your AP exam if they ask you to do it? Yes. Do you have equations to do that, whether it's a cube or a sphere or anything like that? Okay, perfect. All right. Then we gave the example about not only for things to get exchanged across, but thermal regulation. And we talked about things like an elephant that is super huge, right? And how does it regulate its temperature? Because we get hot and we're much smaller than an elephant. What do they use?
Floppy ears.
Floppy ears that are very, very what? Thin. That allows them to lose that heat. And then conversely, looking at another mammal who doesn't want to lose heat, like a whale who's in really, really cold waters, it doesn't want it. It wants to be as large as it possibly can, as thick as it possibly can, so it can conserve its heat. And then we talked about how when we shiver and we're cold, what do? We do this, right? We're decreasing our total amount that's exposed to the area because we're trying to stay warm. All right. Then we went into the prokaryote. And we talked about how prokaryotes can be all different sizes and shapes. There were 3 shapes. You don't need to memorize those anymore, so I digress. When we look, that is so annoying. When we look at prokaryotes, if we want to start from the outside in, and we'll work our way in, okay? The outermost, farthest one thing I can see right is this thing. What is that? And what does that help do? Propel it. Good. Now, what's another way to make it move? Cilia, which is a bunch of short hairs, right? Okay. What are some other attachments out here that might help it stick to a surface? Fimbriae. Say it. Fimbriae. Okay. That helps it attach to a surface and stay put. Okay. Could be in your intestines or something like that. Okay. There are also some external structures out here. One, when the going gets tough and the environment changes, maybe you have some DNA that I would like. What can I use to maybe get some of her DNA or a copy of her DNA? Sex pillows, right? And we can exchange DNA across that sex pillows. And I tend to do that when it's in a crisis, so maybe I'm more adapted. All right? Now, staying out, what is the outermost casing on the outside? Not a peptidoglycan. That's one end. Glycocalyx, right? And it could be hard or it could be what? Slimy. Yeah, it could be hard or it could be slimy. It's a carbohydrate layer, okay? Later, we're going to learn about like gram-positive and gram-negative bacteria and how they're identified, and that's going to come into play. All right? Then go in one layer from the carbohydrates. What's in one layer? What's there? Cell wall. What is that cell wall made out of? Peptidoglycan. Why do you have a cell wall?
To keep structure.
Keep the structure. Do you think water can cross that cell wall? It would die if water could not cross that cell wall, right? Water has to be able to cross the cell wall. If it's impermeable, that thing's going to die, right? Water, what else could cross it? Oxygen, if it's aerobic bacteria. It might be an obligate anaerobe and it won't live in oxygen, just saying. Okay, what's another thing? Carbon dioxide, right, might be able to cross it. So it is a tight lattice, right, trying to give it its shape and its structure so it doesn't blow up, doesn't shrink too much, right? That's your cell wall. It is a protective barrier, gives it its shape. Just inside the cell wall is what? Cell membrane, okay? The cell membrane decides what gets to come and what gets to go out. And remember, we already started talking a little bit about that cell membrane, and we call the cell membrane a what? Phospholipid bilayer. And then looking forward to understand the significance of the cell membrane, peeking into chapter 5, which we haven't talked about, we said, how can things cross the cell membrane? It can go through the phospholipid bilayer, as long as it's not large or charged. Okay? A second way to get across the cell membrane is if there is some channel within the protein itself. So if you're charged, you can still get through to the other side. And the third way to get across the membrane is if you do exo or endocytosis, right? So when we say, I'm just trying to add significance to why we say the cell membrane decides who goes in and out. The cell wall is kind of like a space like this, right? So is this going to prevent water from moving or oxygen from moving or CO2 from moving? No. Okay? But a cell membrane just interior of that is going to be regulating, and that's because we know that the region in the middle of this cell membrane is what? Hydrophobic. And either edge that is either exposed to the cytosol, to the interior of the cell, or the liquid medium that it sits in, it's going to be hydrophilic. Those are the polar heads. Okay? So then we looked at a picture like this, where I'm just going to click it so you have it in case you stop it on the video, or maybe not. Maybe I won't. Okay? Where we tried to remember what all these things do. And one I would really like to point out is one that increases surface area. And what is that called? Mesosome. And the reason why I would point that out is just to remember to talk about how you can have infolding of membrane, and looking further and deeper into this chapter, then it makes us think about what? Endosymbiotic hypothesis. Right? Is this not foreshadowing right here, my English students? Right? A mesosome. I'm foreshadowing the ability to create a eukaryotic cell. Because if I can fold in a little bit, I could fold in a little bit more, and I could fold in a little bit more, and I could be endoplasmic reticulum. I can fold and surround the nucleoid region, and then I can create a nucleus instead of a nucleoid region because I'm surrounded by a membrane, right? So that mesosome is a little foreshadowing. Yes? Okay. Now, where the DNA is, and we didn't talk about that in the previous chapter, where the DNA is located, that is called our previous picture, is called the nucleoid region. It is not surrounded by any kind of nuclear envelope. It's just the area where the prokaryotic chromosome resides. What is the shape of a prokaryotic chromosome? It is circular. How come it doesn't appear circular in this picture or any of the other pictures I showed you? Because it's what? It's all crunched up, right? Like something out of a hairbrush. So it is a circle, but it's all crunched up to fit inside of that cell. Okay? You can have bonus pieces of DNA too. What is that? What are those called? Plasmids, little bonus. And are you going to genetically engineer some plasmids this year? Yes, you are. Okay. Also to the interior of the cell, workbenches for protein synthesis. Say my name, say my name. Ribosomes. Beautiful. Okay. Yes. Good. Good. What was the name of the cell wall for bacteria? Peptidoglycan. Say it again. Peptidoglycan. Nice. So here you can see, now, this is the plasmid. That bacterial chromosome, it looks like it's not drawn to scale. It would be much, much, much bigger than that, and then it's all crumpled up inside. And if I was going to know a bacterium's name, I would know cyanobacteria. Because it not only does it do photosynthesis, but it does nitrogen fixation, and I haven't taught you this yet, but probably was a big proponent of the oxygen revolution 2.3 billion years ago. We'll learn that later. Okay? So cyanobacteria is super, super important. What are you talking about? The oxygen revolution, it changed the entire Earth because it went from an anaerobic environment to an oxidizing environment. Created ozone. So you had a surface, you have protection so life could come out of the ocean. My gosh, how exciting. All right, now, let's talk about, we could take all life is built out of what? What is all life built out of? Cells. Okay, so we can take all life's cells and put them in how many baskets?
3.
Two. Okay, and the name of those 2 baskets are prokaryotic and eukaryotic. So cells are either prokaryotic or eukaryotic, right? And prokaryotic is what we just studied. Smaller, right? No membrane enclosed organelles, okay? Then eukaryotic, I'm going to talk about that again in just a minute. We could take, though, all the life on this planet and put them in 3 baskets, okay? What are the 3 baskets for all living organisms?
Eukarya, archaea, bacteria.
Okay, bacteria, archaea, eukarya. I'm saying it in that way because I'm doing it in the order of evolution, probably. Bacteria, and then archaea, and then eukarya. Now, remember, archaea is misnamed because arch means ancient, but it's really not ancient, right? They thought it was ancient, but it's not. These are archaea, are the extremophiles. They live, thermoacidophiles live where it's hot and low pH. Halophiles live where it's salty. Methanogens live in intestines. So those are extremophiles. They have different cell membranes. Their cell membranes are not made with fatty acids, but they're made with hydrocarbons. What else?
They live in extreme environments.
They live in extreme environments and their cell walls are different. It's a polysaccharide. It's not peptidoglycan. And we're going to learn later, they have a little bit different ribosomes. And are archaea really closer to prokaryotes or closer to eukaryotes? That's right. And they're really closer to eukaryotes when you look at their DNA or when you look at their ribosomes. And so even though they were given that ancient name, archaea, they are not actually ancient. Now, what do archaea and bacteria have in common? By definition, they are both what? Prokaryotes. Okay? Yes?
So I know people don't say viruses aren't bacteria. Are viruses considered?
Viruses are not alive.
Oh, they aren't?
No, they are not alive. And we're going to talk about them. but they can't exist on their own. I don't mean just like a parasite, but they have to use a totally different organism, and we'll talk about that. They're not self-replicating. They have to live within another. Okay, eukaryotes we can put in 4 baskets, but again, it depends on what book you're looking at and what classification system you're looking at. So when you take all eukaryotes, the simplest of all, you're looking at protista, generally unicellular. They can be autotrophs or heterotrophs. Plantae are primarily multicellular, and they are autotrophs. Animalia are always multicellular, and they are heterotrophs. And fungi are primarily multicellular, and they are also heterotrophs. Okay, and we'll be studying each of those, but those are under the kingdom. All of these organisms are made out of eukaryotic cells. So then the question that we had is, where did eukaryotic cells come from? How did they evolve? Okay, and so when you look at the evolution of eukaryotes, I would focus on 2 things. One I would focus on is the energy organelles. Who are the 2 energy organelles? Mitochondria and chloroplast. They both have items that are just like free-living prokaryotes. A photosynthetic prokaryote and a chloroplast are very, very similar. And aerobic bacteria are very, very similar to mitochondria. And some of the evidence we have is that both of them have a what membrane? A double membrane. Okay? And the outer membrane is like a eukaryotic membrane, and the inner membrane is like a prokaryotic membrane. How did it get that second membrane? Maybe because it was engulfed, right? Okay? So now we have 2 membranes. Also, they each have their own DNA, and that DNA is in a circle, just like prokaryotes. They have their own ribosomes and can make their own proteins, and their ribosomes are just like the ribosomes of prokaryotes. Okay? And they are self-replicating. When you need more, they can make more of themselves. Now, they have abdicated their self-control. They can't leave a cell or anything like that, but that has been given up to the nucleus of each of those eukaryotic cells. The second piece of evidence I would look at is that potentially the nuclear envelope and all the membrane-enclosed organelles, besides the mitochondria and chloroplast, could have been generated by the outer membrane folding in on itself, and thus forming a little highway. And it could have gone around a mesosome that was just a really big mesosome, could have gone around the nucleoid region and created a nucleus. And lo and behold, the nuclear membrane, the nuclear envelope of eukaryotes is not a single membrane, but a double. So it actually has 4 layers of phospholipids, right? Follow? Okay? And then the ER would just be an extension of that, and lo and behold, when you study ER, it is continuous with the nuclear envelope. So it's like that membrane folded in, went around the nucleus, and just went yeah. And some broke off and became the Golgi apparatus. Okay, and then as proteins were synthesized, some destined to be in the cell, like a lysosome, or some proteins destined to be exported out of the cell. Those could be just packaged in that same membrane, creating all those vesicles we have. It's just repeating that same thing. So that is the endosymbiotic hypothesis, the origin of the eukaryotic cell. All right, so let's try this again. And let's see if we can figure out who would we say is each one of these. Who would be the doorman? Yeah, I would say the cell membrane. Who would detoxify? They could, so too could the smooth ER. Okay? Remember we talked about the role, and we talked about don't be an alcoholic because it'll ruin your smooth ER? I mean, don't be an alcoholic for many reasons. But one is it will ruin your smooth ER. Okay? So there's a couple in there. Who supports? Well, it does not have a central vacuole. If you were in a plant, it could be a central vacuole, right? If you were a plant, you could also have support from your cell wall, but also on support the cytoskeleton. Yeah, if you can see the underlying cytoskeleton. Who is the UPS man? Golgi apparatus, and here it is. And the part that faces the nucleus is called the what part? cis, and the part going away is transporting on out. Okay, maybe how you can think about it. Who is our energy organelle? Exactly. Who stores? Vacuoles. Any one of these could be a vacuole. Let's make that a vacuole. Protein synthesis. Now, some ribosomes are anchored on a membranous structure. Who are they anchored on?
Rough ER.
On the endoplasmic rough ER, endoplasmic reticulum. And so that's called rough ER. Some are free. They're just floating around in here. Okay? Who has ribosomes? Rough ER. Who is the brain? Nucleus. Who digests? Lysosomes. Okay? The stomach of the cell. Remember the churros? Oh, we don't know about the churros, right? Remember the churros are the centrioles. There's two of them. They're perpendicular to each other. They have they have 9 sets of 3. And then when we looked at cilia and flagella, they have 9 sets of 2 with 2 in the middle. So we're like kind of similar. And the sets of things that we have are those microtubules. That would be part of the largest part of the cytoskeleton. But then the connector between those, who anchors the flagella or the cilia? Basal bodies. And those basal bodies are just like centrioles that have 9 groups of what? 3. And that was one of the last. Okay, here. Do you remember that? Yeah. Okay, so that was that. All right, so let's keep going. When we looked, let's look here, at the plant cell, we look at things that are unique to the plant cell. What is something unique to the plant cell? Tell me. How big is the vacuole? Large central vacuole. Because it fills up with what? And it pushes against the cell walls. And by doing that, that's called turgor pressure, because the walls are going to push back. It keeps the plant rigid so it can do what process? And if you don't water your plant, it wilts and it can't. And then you water your plant, it fills back up again. Okay? Now, other organisms can have vacuoles too. Like a protist can have a contractile vacuole, and it's all about water regulation, right? It's squirting out water because water keeps coming in, and we'll learn about why water would come into it, but it keeps squirting it out. It could be a little storage vacuole in like our animal cells. But a big, large one like this is like the multi-purpose room, okay? What else is unique? Cell wall. What is it made out of? Cellulose. Good. What's a fungal cell wall made out of? You're so smart. What else is unique? Chloroplast. Okay? So do chloroplasts do plants do photosynthesis and animals do cellular respiration? No. Who does cellular respiration? All cells. Yeah, all cells. Okay? All right, good. Very good. Is there something unique to an animal cell that a plant cell doesn't have? You both have mitochondria.
The churros.
Yeah, lysosomes. The churros, the centrioles. Okay? All right. Now, let's focus in on the nucleus. We already know it's a double membrane, and we already know it encloses the DNA. Now, there's a central structure inside of it. What is that called? Nucleolus. And what gets made in the nucleolus?
Ribosomes.
Don't say that. Tell me something else. RNA. That will make ribosomes. Ribosomes consist of protein and rRNA. So where you're making all that rRNA is right there in the nucleolus. Just speaking of ribosomes, they have 2 components. They have a
small
and a large. Okay, good. Okay, and when you look at the cell, most of the time it's in what's called interphase, and all the DNA just looks all scattered, and it's called chromatin, and it's a mixture of DNA, primarily RNA and proteins. But when it gets ready for cell division, then the DNA starts winding up on these protein spools called histones, and it will form these rod-shaped chromosomes. But you only see that when it's getting ready to divide, where you see these rod-shaped chromosomes. Surrounding the nucleus is the ER, rough and smooth, right? And on the rough ER, what makes it rough are and proteins can get made on them, right? And then those proteins will move into the interior of the endoplasmic reticulum, and now it's like freeway ride. It might get modified. Out here in the smooth ER, its job is to make more lipids and to detoxify. That is its job. When you move from ER, what is your next destination? Golgi apparatus. And here's just a little peek at that. Okay, stop. Okay, and then Here you can see small and large ribosomal subunit and the protein moving into the ER. And this actually is my favorite picture here. That's a good one in your book. It's good English. So you can see the nucleus, you can see the rough ER, you can see proteins going to the cis side of the Golgi, you can see packaging of lipids and going to the Golgi. The Golgi can send it for export, exocytosis, and then this is a good time to talk about the stomach of the cell. The stomach of the cell is called the lysosome. Good. So be a lysosome. I've engulfed this bacteria or this food, and I am just an endocytotic vesicle. I can join with this primary lysosome. He's now a secondary lysosome. Digest, absorb, and then we can go to the edge of the cell and get rid of anything else we want to get rid of. So lysosomes move through the cell. Okay. Now, how do all these vesicles move? Remember Walking Man? Okay. So Walking Man, it's not like, oh, I ran in here. Hey, I'm a nucleus. It's not like that. The cytoskeleton is supporting all the structures that you have within here. All right. And then that brings us to here. Okay. And then remember I told you, I'm sorry you have to learn some of these a little bit early, but that's how the new, the 2020 test is going to be when you do your self-check. So let's review a chloroplast, all right? First of all, it's got a double membrane. Its double membrane is right here on the outer part, but then the inner membrane is called a thylakoid membrane. Say it. Thylakoid. Say it again. Thylakoid. And on those thylakoid membranes, when they're stacked up like green pancakes, and they're hollow green pancakes, very light and fluffy. But on those hollow green pancakes, that's where the chlorophyll is embedded, and that's where you do the light-dependent reactions. And that's where a plant can harvest light energy and turn it into chemical energy. The two pieces of chemical energy, this is photosystem 1, photosystem 2, we're going to be talking about that, but it makes ATP and reduced NADP. You do not have to memorize those words yet. I just want to use the right ones. Out here in the stroma, surrounding the green pancakes, is where you do the light-independent reaction. It used to be, when I grew up, called the dark reaction, but that implied that it needed to be done in the, and it's not that it needs to be done in the dark, you just don't need any what? Like, this is where you're actually building the sugars, where you're fixing the carbon, and you're going to use the ATP and the reduced NADP from the light reaction and CO2 in order to build those sugars. So the sugars get built, and that's called the Calvin cycle, out in the stroma. Okay? That's good. Yeah, that's good. All right. Now, a chloroplast is part of the bigger grouping of organelles called plastids. If it's got any color in it at all, it's called a chromoplast. Chromo, how can you remember that means color? Chromatography? No, that's science.
The chrome symbol.
What?
The chrome symbol. It has color on it. Like
Okay.
Google.
Sure. Oh, yeah. Google Chrome. It does have color on it.
Yeah.
Okay, I like it. Now I got it. And that is misspelled. That is leucoplast. That's where you store carbohydrates. All right. Energy related. Mitochondria was also, I don't know why I repeated that. So you have an outer membrane. Okay, on the outside. The inner, you just have one inner membrane, and that has a bunch of folds on it, which increases surface area. And it's here on these cristae, this inner fold, that you're going to do the electron transport chain, which ultimately is going to make a bunch of ATP. Now, there's several other reactions that occur. The Krebs cycle occurs inside the matrix of it, the very center. I can't remember how much I told you about that, but there are different, the electron transport chain occurs on the cristae, the Krebs cycle is in the interior. That would probably be the only two I would know on that. Again, it's going to be a lot easier for you to remember that once you understand those reactions and we do them. It'll make more sense, and structure does equal function. All right. Next. We talked about the cytoskeleton. You don't have to memorize the different parts anymore, but to really understand kind of cell division, I want you to make sure you understand about these microtubules. And if you remember, the centrioles come out of the microtubule organizing center, And so all of these microtubules are going to come out of that, and that's going to help to divide the cell. But plants don't have centrioles, and they do division just fine. We don't know why they are there, but they are there. Again, could be related out here to the basal bodies. Okay. Why don't you, with somebody next to you, and do it quietly, you have to name what I'm pointing to and tell what it does. Okay, and just ping pong real quick. We'll do it real quick, back and forth with somebody next to you. So, do all the arrows, and then I will ask you what they are. Go ahead. You can do this at home, too. It's good for you. Okay, pause. Let's do a quick self-check. What is this pointing to right here? Mitochondria. Mitochondria. Good. What's this pointing to? Central. Good. What's this? What's this? Rough yard. Good. What's this? What's this? What's this? Nucleus or nuclear envelope? What's this? Cell membrane. What's this? Did I hit them all? Okay, good. All right. Go again. Hit the arrows. Okay, stop. Come with me. What's this? Cell membrane? Nice. All right. Again, I recommend find some diagrams where they're also labeled, you know, with structure and function. And here, why don't we go in as something, walk out that door. Walk out that door. Don't turn around now. Let's go in as something we're looking forward to this weekend.
It says the website is temporary.
There we go. Just put your all in here right now.
Oh, the Wi-Fi is not.
Okay, they're all logging in. I'm going to click through the questions so you can see them too if you're at home watching. I hope you're having a good weekend. All right, peeps, we're going to start. You can still get in.
Oh.
It's not letting you in yet?
What should we join as? That's a game character. That's a game character.
Oh, okay.
That wasn't me, but.
Playing Minecraft? Okay, I'm going to show you each of the questions one at a time. I recommend that you get out a piece of paper and commit to your answer. And then when we go back through again, you can check to see how well you understood it. So I'm just going to slowly click through them. So write down your answer, and if you feel like you need more time on any one question, then just hit pause on your recording. Number 2. Number 3. Here comes number 4. And number 5. Commit to your answer. You can do it. I believe in you. Number 6. And number 7.
Oh, that's the bad one. What's the other one? That's the
Number 8. 9.
What?
Number 10. Number 11. And number 12.
And then this one is
And number 13. Now I'm going to click slowly back through them, double check your choices, see if there's any you need to change.
I know
I think we've got extra names.
people forgot to walk out that door.
They did forget to walk out the door. I think 66 or 67 is 100%. All right, peeps, are you ready?
yes.
All right, here we go. Let's check it. Yes, I would like to end in review. Oh, powerhouse of cell, mitochondria. Guys, look at me for a minute. Guys, please look at me and stop. Stop. You may have gotten it right, but the person next to you may not. So please let them listen as I go through each one. Plus, we've got some people at home. Say hi. Hi. That are trying to go through their answers as well. So which organelle is the powerhouse? Mitochondria. Nucleus. What would I say for the nucleus? Not the powerhouse. It is the brain of the cell. Okay. Number 2. Almost everybody got this. Who goes in and out? Who decides? Cell membrane. Through the phospholipid bilayer, right? Or through a protein or endoexocytosis. I'm guessing they guessed cell wall. They did. 2 people did. Cell wall doesn't decide. It's just like this lattice-like barrier. If you can fit through, you can go through. Okay, and what else did they pick? Golgi apparatus? Not even a little bit. You're way off the mark. Okay. Number 3, which organelle processes, packages, and distributes? This is our UPS man. Okay, because it is the final distribution point for export or to keep inside the cell. That's your Golgi apparatus, not your ER. J, ribosomes. No. What happens on a ribosome?
Build proteins. Workbench.
Okay. 4. Good job. Everybody got it, except for these 28 extra children. Supports, protects, is permeable to gases. That's your cell wall. Not, what did you say, cell membrane? No. Is the job of the cell membrane to protect? No. Okay. Is it to support? No. The cytoskeleton, if you don't have a cell wall, would do that job. Yay, control center nucleus. Yay, ER compartmentalizes. Yay, on the chloroplast.
No.
Oh. Which organelle is the multi-purpose organelle? Stores, transport, support, trigger pressure plants. Vacuoles. Might have, no. Which structures? Yes, all 53 people who logged in got that. Okay, what would be found in both plant and animal cells? Mitochondria. Do you have a cell wall? No. Then you're not going to be found in an animal cell. Logic. Which of the following has 9 sets of 3? Centrioles and basal bodies. Has 9 sets of 3. Okay? And then number 13, who has 9 sets of 2? Cilia and flagella. Okay. You guys are awesome. Have a good weekend. No sex, no drugs, no alcohol. What happens in Vegas?
Stays on your feet.
Oh, no.
Are we allowed to say that?
Yeah. All right, make good choices. Make good choices.